Did genomes increase in size smoothly over the course of evolution? Why did sexuality arise, and what evolutionary advantages does it have? We will try to answer these and similar questions in today's episode of the series Secrets of Genes, in which I welcome you. My name is Edward Kejnowski, and I work as a geneticist at the Institute of Biophysics of the Czech Academy of Sciences. Evolution mostly took the path of genome enlargement. Not always, but most of the time. You might be wondering why that was the case. Where is this drive toward larger genomes encoded? Is it inscribed in the genome itself, or rather in the cell? Why do organisms tend toward greater complexity? Is the tendency toward higher complexity a property of matter? After all, there are known exceptions, such as the genomes of parasites, which have shrunk over the course of evolution. And how is the complexity of the body structure of plants and animals related to the size of their genome? Recent research suggests that neither genome size nor gene count evolved smoothly over time, but rather that there were certain jumps or temporally limited genome expansions. The causes of these changes and their mechanisms are still shrouded in mystery. In the previous episode, we talked about qualitative leaps in evolution. We discussed the transition from prokaryotes to eukaryotes. Another significant leap was the emergence of multicellular organisms. This occurred after roughly 3 billion years of evolution, during which life existed only in a unicellular form, and it happened independently in many lines of organisms. This allowed for the emergence of various specialized cells and the division of labor among them. Although the genome of all cells in the same individual remained identical, gene expression differed in various cell types according to the function of each cell. Genes are switched on and off in individual cells according to a complex plan for the development of the organism. There is complex communication between cells. Another remarkable evolutionary transition was the emergence of sexuality, and with it the coexistence of two copies of the genome in eukaryotic cells. What do I mean by that? In our cells, we have two sets of chromosomes, one from our father and one from our mother. Both sets of chromosomes are very similar, but they are not completely identical. Only our sex cells, which fuse during fertilization, contain just one set of chromosomes, because otherwise the number of chromosomes would double with each generation. Cells carrying one set of chromosomes are called haploid, and cells with two sets are called diploid. The alternation of haploid and diploid phases occurs in all sexually reproducing organisms. Different species or entire taxa differ only in the duration and morphological visibility of both phases. The alternation of these two phases can already be observed in some unicellular organisms. In multicellular organisms, the bodies are made up of diploid cells, while haploid cells are practically invisible. In lower plants, the haploid phase is more distinct, but it is still dependent on the diploid phase. The transition to sexually reproducing organisms that alternate between haploid and diploid phases can therefore be considered a significant evolutionary milestone, which occurred repeatedly in different taxa due to its advantages. Sex is often determined by external factors, such as temperature or even population density, but in many cases it is genetically determined through sex chromosomes. These are referred to as X and Y, or as Z and W. You probably remember from school that men have XY sex chromosomes and women have two X chromosomes. The main advantage of sexuality is the ability to continually remix that genetic cocktail of genes and quickly eliminate undesirable, harmful genes through the process of recombination. Sexuality gave genomes an entirely new way to rearrange their modules that have proven themselves evolutionarily, and to create new qualities. It became possible to use an astonishing number of combinations to create new functions, just like when we build a small house out of Lego one time and a large castle another time. So much for the biggest leaps or transitions in evolution. Besides these, however, we can also observe changes of smaller scale and significance, even within individual taxa. Both abrupt and gradual evolution occur in all organisms. However, gradual evolution is more typical of asexual unicellular organisms, while evolutionary leaps are more often observed in multicellular organisms that reproduce sexually. The same holds true for their genomes. Bacterial genomes are small, simple, compactly organized, and efficiently utilized, and they evolve through the accumulation of small mutations. In contrast, the genomes of higher organisms are large, contain many repetitive DNA segments, so-called repeats, and their evolution often involves major, large-scale rearrangements, such as the duplication of the entire genome, known as polyploidization, or the transfer of genetic information between species, called horizontal gene transfer. That's all for today. In the next episode, we'll talk about what the genomic landscape of an organism looks like, and what forces drive genomic expansion. I'm really looking forward to seeing you. If you enjoy my storytelling, I would appreciate a like. And if you'd like me to continue making episodes about genetics and evolution, please subscribe. That will help these videos reach more people. I really appreciate your support. Thank you, and bye-bye.